Chapter 4 video lecture on the cell. All body processes depend upon the cell. Likewise, the cell is the basic structural functional unit of the body. So the cell is the basic structural functional unit of the body. I do not care for this slide. 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 Now on this slide, it is a rough idea that you need to have. You should have an idea of roughly what the size of a red blood cell is, the size of a human egg. You need to have some idea of dimensions of size. You also need to know, as you will for this chapter and the next, that cells do come in different shapes. They come in a spherical shape, a cube-like, sometimes called cuboidal, column-like or columnar, cylindrical, disc-shaped, or even irregular wonky shape. So be aware that the size is of some variable and some shape is of some variable. This is the picture that you need to have in mind. You do not have to memorize it, but you should have some idea of the relative sizes. So you know that a human egg is not as small as a virus, right? So a human egg is definitely bigger than a virus. So some relative idea is what you need to have. Okay? Like for example, there are many air filters out there that say that they only protect against bacteria, but the air filters don't protect against viruses. Well, the one that I'm using for COVID-19 that I use as a added filter inside my little inlet or inslet that I put inside of my mask, that one is an air filter I use that actually works with viruses. Most air filters don't. The more expensive ones actually have a degree for virus protection as well. So that's what I'm getting at. You should have a relative idea of the sizing of these things. Here are the shapes. Again, I don't really care for that picture. Okay. Now you should also know that the cell has an outer covering called the plasma membrane. Some people call it the cell membrane. Same thing. Now remember, most of the cell membrane or plasma membrane, and I say most meaning like 85% of it, is phospholipid. You learned that in the previous chapter. So 85% of the plasma membrane will be phospholipid. You also learned that the cell membrane has some things like cholesterol built into it for flexibility and bendability. So you have cholesterol inside the cell membrane, and you have phospholipid primarily that make up the cell membrane. Keep in mind that the cell membrane is a barrier, but it does also have some allowances for certain things to go through and not. So I mentioned before, lipid-soluble items can go through the cell membrane, whereas water-based items, water-soluble items can't easily get through. So that's the kind of knowledge they're expecting with this slide here from the past. So the cell membrane is a nice barrier, but it does allow certain things to come in and certain things not. That's where I mentioned previously in a previous slide, aquaporins. So aquaporins are as a water channel, right? Allowing water to get through. So that's the kind of thing that you need to be aware of. Then we have our nucleus down there in the middle there. That is where your DNA will be. So the nucleus will be where the DNA is. Then we have our cytoplasm. Now cytoplasm is this big open space inside. So if we take away the cell membrane, we take away the nucleus, and all you're left there is cytoplasm. Now inside the cytoplasm you have a liquid. That liquid goes by the name of cytosol, which you see at the very bottom. Also embedded in this cytoplasm are organelles. Okay. So now you see the term I spoke of, which is cytosol, which is the liquid, the viscous fluid. Then we also have our organelles that I spoke of. Now the organelles come in two categories, as you see at the bottom there. They can be membrane-bound organelles, or they can be non-membrane-bound organelles. So we'll see that a little bit further as well. Okay, so this is what I've done so far. Cell membrane, nucleus, cytoplasm. Now these are the organ-bound or membrane-bound organelles. You will need to know this list. So this list is of value to you. So the membrane-bound organelles will be the Golgi, the endoplasmic reticulum, the lysosome, peroxisome, and the mitochondria. Those are membrane-bound organelles. The non-membrane-bound organelles would be things like ribosomes, cytoskeleton, centrosome, and proteasomes. Now, Nimish, do I need to know what these are as you go through this chapter? Yes. The suggestion I'm making now, and I'll make it again in a few minutes, is make sure you put them on a flashcard. I have never seen a student succeed on knowing the organs or the organelles of the cell unless they put it onto a flashcard. So the image, one side, information on the back side of the flashcard. That's the best way to do it. So here you have again the membrane-bound versus the non-membrane-bound organelles. Then we have the inclusions. I don't really care too much about those. And here's the structure of the cell. This is what I mean by putting it on a flashcard. You need to go around this picture like a clockwise fashion and put these items onto a flashcard. That is the way you're going to understand the functions of these items. Now I'm going to take a cursory look and giving you an overview, but you still need to take the task of making flashcards of this image, structure, and function. Okay, we'll start at the top. We have the first one here called rough ER. It is called rough because it has the ribosomes attached to it. So rough ER has ribosomes attached to it. You can see it in the picture with these little circles. Now the rough ER plays a role with protein synthesis. So rough ER will play a role with protein synthesis. Now there's a catch. You will see that there's two kinds of ribosomes. I mentioned a moment ago that rough ER has bound ribosomes, but then there's a separate entry here that says free versus fixed. That's what they're describing here. So the fixed ribosomes are the ones that are stuck to the rough ER. That's why you see these labeled as fixed, but they're really part of the rough ER. Now what is the difference? The fixed ribosomes deal with protein synthesis, as does rough ER, deals with protein synthesis, but it's for export purposes. So the proteins that are made by way of the rough ER will eventually find its way outside the body. And when I say outside the body, I'm talking about the cell itself. It will eventually end up in the bloodstream and go elsewhere in the body. So rough ER, through its fixed ribosomes, will make protein for exporting out of the cell. Well then, Nimish, what about these free ribosomes that are all clustered everywhere? The free ribosomes are making proteins for the cell's internal use. So free ribosomes are for the cell's internal use when it comes to making proteins. That is a big difference. Next structure is smooth ER. Smooth ER is for lipid and carbohydrate metabolism. So lipid and carbohydrate metabolism. And that'd be the smooth ER. These are the ones that have no ribosomes attached to them. That's why they're called smooth. So smooth ER is for lipid and carbohydrate metabolism. The next one down is mitochondria. This is where ATP is produced. So ATP production. The next one after that is the Golgi apparatus. That's this thing right here, the Golgi. Now it has an input on one side and an export on the opposite side. Input and export. Now the way it works is it takes information from the rough ER Then it takes it to the Golgi. The Golgi's job is to package, sort, and modify the protein. So it's going to sort it, it's going to ship it, it's going to package it, it's going to modify it, and then it's going to export it out. That's what the Golgi does. The next one after that is peroxisome. This contains hydrogen peroxide. So this will contain hydrogen peroxide. It is the same hydrogen peroxide, should it ever get loose, that will end up killing the cell. So some people call it sudden cell death, and that's because of a bursting of the peroxisomes. And below that we have lysosome. Lysosome's job is to contain digestive enzymes. When things come into the cell, they may be too big for the cell to process. So they have to be put into digestive vesicles, which are called lysosomes. So lysosomes contain digestive enzymes that will break things down. Then moving around the clock here, we have our cytoplasm, which I said is all this fluid environment containing the cytosol. So the fluid environment containing the cytosol. Here's the plasma membrane, the outer wall. Cell membrane, plasma membrane. Remember, it's 85% phospholipid. It has cholesterol embedded in it for sturdiness or steadiness. Okay. Then we have our microvilli down here, which I don't care too much for. Then we have our cilia and our flagellum. Now, the cilia and the flagellum really belong to something on this side called cytoskeleton. And under cytoskeleton, which I'm over here now, cytoskeleton, it contains microtubules and microfilaments. So microtubules, microfilaments. Now, are you telling me that the cell actually has sidewalks and highways and roadways? I always thought of it as a big blob that things just, luckily got there somewhere through tides and grace. No, the cytoskeleton is actually a pathway, a sidewalk, a road, a tram rail, whatever you want to call it. There's actually roadways and pathways and highways within this cell, and they're made out of cytoskeleton, which is either microtubules or microfilaments. So microtubules, microfilaments, and these are the things that give it some sturdiness. They also give it some way of moving things and transporting on, like on monorails or on tracks, like sidewalks or roadways. Okay. Then proteasomes, again, they're just something that contain protein enzymes, so proteasomes. Then we have centrosomes. Now centrosomes are going to be part of something called centrioles. So centrosome is part of something called centrioles, and you can see one right here. That is going to play a role in cell division. So when the cell is ready to divide, the centrosomes containing centrioles will help with cell division. Okay, we've already taken care of the fixed ribosomes, free ribosomes we've taken care of. Here's our nucleus. There's our DNA inside of that. And this is where you're going to have your 46 chromosomes. So 46 chromosomes for the cells. Okay. Then down here, the bottom, which I haven't talked about, are the vesicles. A vesicle is simply a little tram, a little car, cable car, if you want to call it, that takes you from point A to point B within the cell. So just like a cable car is actually running on some cable, this vesicle is actually running on some type of cable. Remember I said cytoskeleton cable that allows the vesicle to go from point A to point B. If you ever ride those rides at Disney, they're actually on some kind of track, even though it may not seem like it, right? So even the Small World After All ride is on some kind of track. Even though it's on a little mini boat, it is still on some kind of track. Okay, so that's a vesicle. It takes you from place to place within the cell. So again, make sure you have a more clarified view of the function of these items. Okay, this is the general thing that a cell does. It maintains the shape of the cell, integrity of the cell, gets nutrients from the outside, disposes of waste, has cell division. That's why I said centrioles play a role with cell division. Okay. These are the things that we've already talked about when it came to the cell membrane. We said that it has phospholipids. Remember, 85% of it's phospholipid. It's got cholesterol built into it. So this slide is worthy of keep in mind that the cell membrane is selectively permeable. That's the magic phrase you'll see on a test somewhere. It is selectively permeable. It allows certain things and it does not allow other things. Now, on what basis do you typically allow things in? Well, here's the rule, and there's 4 of them. Size, so the size of the item matters, so size. Charge, so the charge of the item matters. The outside charge tends to be positive. The inside charge tends to be negative. So if the charge of the item is positive, it's more likely to want to get into the cell because the inside of the cell tends to be negative. Okay? So size was number 1, charge was number 2. The next one is presence of a channel or carrier. So there must be a carrier or channel that allows for it. I've already mentioned aquaporin. Well, aquaporin is a channel. It allows for water to go in and out. Had the aquaporin channel not been there, then water would not be able to go in and out. So that's what I mean by having a channel or a carrier. Number 4, and the last one, is lipid solubility. Since most of the cell membrane is phospholipid and like dissolves like, that means oily things, and I mentioned this before, oily things, greasy things, lipid things are more likely to get through. Whereas water-based materials like water-based hormones and things like that you'll need to know for A&P2, well, guess what? They can't get in. They would need some other technique. So again, the 4 characteristics of what is allowed in or not. Some people call it parameters, characteristics. Well, the 4 things that go under selective permeability, the 4 things are size, charge, presence of a carrier or transporter, and lipid solubility. Okay, next slide. Okay, so here's our phospholipid. Again, I've said this so many times, so we can move on from there. Cholesterol, I've already told you, it adds strength or stability to the cell membrane, so I've already mentioned that. And you've heard me say it several times, so cholesterol is in the phospholipid bilayer for stability and strength. Here's a picture version. Many books have it. And so many books show the phospholipid bilayer. They show the proteins going in and out. They show the cholesterol. There you go with the little yellow. Those are 4 ringed structures. Remember, 4 rings make up a cholesterol, and cholesterol is the basic building block for steroids like testosterone and estrogen, previously mentioned. So that's what I'm getting at. This is your cell membrane. And then down here in the legend, it says selective permeability. And that's what I gave you a moment ago. It's based on those 4 things: size, charge, lipid solubility, and whether there's a carrier or transporter for it or not. Now the next thing I want to go over using this picture, because that's what we're going to head over into, are these things, sticking up and sticking out. Now there's 2 categories of these. These are called membrane proteins. So these blue things are called membrane proteins. And there are 2 categories. We have integral protein and we have peripheral protein. The integral protein would be like this one here that goes right into the cell membrane. Okay, that's the integral kind. The other kind is peripheral, where they are either just on the inside or just on the outside. So sticking just on the outside or sticking just on the inside. They are peripheral. So we have 2 kinds of membrane proteins, either integral or peripheral. Now, what do they do? We'll see that as a long list in a little while, so I'll hold off on that. But we have membrane proteins embedded within the cell membrane, peripheral or integral. Okay, next slide. So here's what I was speaking of. So here's what we have so far from what I've just said. Membrane proteins come in two varieties. We have either integral or peripheral. Okay. Now, next slide. Again, we have integral and peripheral. Nothing new so far. Okay, so what I've said is this picture. We have integral and peripheral membrane proteins, and the functions are about to come up. Okay, these are the functions. We're going to go line by line on this one. The first function is transport or carrier. I need to say both of them because it depends upon the book. So some people call it a transport, some people call it a carrier. You can see the word carrier right down there, and that's why they say that. So a channel, a carrier, a transporter, they're all in this category right here. So channel, carrier, transporter, they all belong to this first category. So they're going to help you move things here and there. That's what these are for. So a channel, a carrier, or a transporter is to move things onto one side of the cell or the other side of the cell. That's how I look at it. Then the next one is called cell surface receptors. Okay? Cell surface receptors. Okay, now what is that about? You have to have receptors somewhere on your body to make a response. Your cells have to have receptors as well. Okay. So your nerve cells, for example, provided here in this example, the nerve cells have to have receptors. Your muscles have to have receptors. Okay. So receptors are both inside your body and on the surface of your body. That's how you're going to have a response. Remember in the first chapter we talked about receptors, then we had the control center, and then we had the effectors. That was in the first chapter. Well, same way here. We have to have receptors, even on the cell surface. Right now, what are we responding to by making antibodies against COVID-19? Well, we're responding to the surface markers on the COVID virus. Same way here. We have to have receptors. Then the third item is cell ID. That's what I call the third one down here. Cell ID. Okay, cell ID. Every cell of your body has a marker that identifies it as you. Let me give you an example. When you were in daycare, preschool, daycare, and you went to school and you took off your little jacket and the teacher there took your jacket off and put it on a hook, or you had your little lunch box and they put it away somewhere and then they took it out of a little cubby during lunchtime. All of these things had what on it? Your mommy put your name on the jacket. Your mommy put your name on the lunch box. Same way here. Every cell of your body's got to have some kind of marker on it. Okay? For example, red blood cells. And you'll learn this in A&P 2. Red blood cells live only about 120 days, when after that the immune system comes in and destroys the red blood cell because it's beyond the 120 days. How does it know that the 120 days are up? Well, the marker must be fading away, and now it goes from this is Nemesh's red blood cell to, since the markers are gone, this is a foreign red blood cell. And now the immune system will come in and destroy it. So that's what I'm getting at cell ID. And unless you have a really, really close identical twin, you're not going to have the exact cells everywhere. So that's why when someone gets a skin graft, it has to come real close. So when you get an organ from somebody, organ transplants, they get the organ cell ID markers really close. But what do they still have to take even though they got it as close as they could? They have to take immunosuppressive drugs to hold on to that new organ because the immune system will still consider it as foreign. And you have to suppress your immune system to hold on to the new organs. That's what I'm getting at. Now, if you had your own body and you needed to transplant cells from your own body, that's not a problem because the cell ID markers will match. Right? So these are the three so far. Okay, continuing, the next one are enzymes. So enzymes are also something to be aware of. Proteins do play a role as enzymes, so therefore you have enzymes. The last one for me is this one, anchoring. Anchoring, that's my last one. Now, anchoring. Have you heard of barnacles? Barnacles are those little sea creatures that stick to the bottom of the ship, and after some amount of time you have to take the boat into dry dock where it's basically out of the water, and then you got to take chisel and scrape them all off. That way they're not slowing down the boat because they're all stuck to the side of the boat. Well, the same way here. Your cells have to stick to something. So the cells that stick to the wall of your intestines, for example, you have your intestines, well, then you must have these cells that stick to the wall of the intestines. Well, how do they do that? They do that here, anchoring sites. So that's another function of membrane proteins. They actually stick to the walls of the intestine. Okay. All right, so those are the membrane proteins. Okay. You will need to know these functions. You will see it. Here is a picture version that many textbooks have. So there's your transporter, there's your receptor, there's your cell ID. That's why these have to match perfectly. There's your enzyme, and my last one was cell anchoring here. So enzyme and then cell anchoring. That's my last one. Okay, this chart is of great value because you'll need to know what we call passive transport and what we call active transport. Let's do the passive transport first. Passive transport would be things like diffusion, osmosis, simple diffusion, facilitated diffusion. I'm going to stop there for a second. So these are all passive. Why are they passive? There are two things that make it passive. One, they don't require ATP. So things that are in this brown here are passive, do not require ATP. Second, they always go down their concentration gradient. That means they are going from high to low concentration. That's what passive is, from high to low concentration. You will see the language on a test that says down their concentration gradient, from high to low. Okay, now I'll give you a quick example of these. Diffusion is like when you put food coloring inside a glass of water. That's diffusion. Okay, food coloring in a glass of water would be diffusion. Then we have osmosis. Osmosis is passive. It is simply the movement of water from high concentration to low concentration with a selective permeable membrane. That would be a definition to remember. So osmosis is the movement of water from high concentration to low concentration with a selective permeable membrane. Then we have our simple diffusion. Then we have facilitated diffusion. Now facilitated diffusion still goes from high to low, but it does require a transporter or carrier, even though we're going from high to low. I look at it as moving sandbags from a high pile and creating another pile on the other side of the cell. So taking a high pile of sandbags and giving it to somebody on the other side to create another pile. You're going from high concentration to low concentration. That's selective. That is facilitated diffusion, and that does require nothing more than a transporter. Okay, so one more time, moving sandbags from high to low. From one side to another, that would be facilitated diffusion requiring a transporter. Okay, I don't care for these two down here. Now we go over here to the active site. Active does require ATP. So active requires ATP, but it goes from low to high, which the books call it against the concentration gradient. Okay, so they go against the concentration gradient. That's from low to high. Here they call it up its concentration gradient. That's the same thing. So up the concentration gradient is going from low to high against the concentration gradient. And it does require ATP. Then on this one here, the active transport over here, same thing. We have the usual kind, primary, secondary. We have things like symport, which moves two substances in the same direction. A very good example of symport that you'll see in the future is glucose and sodium. So wherever sodium goes, glucose follows because that is a symport. So as I move glucose in one direction, let me back up. If I'm moving sodium first. If I'm moving sodium in one direction, then glucose will follow. That's called a sodium glucose symport. So sodium goes, glucose follows. And you'll see that a lot in A&P too. So wherever sodium goes, glucose follows. That's symport. Then we have antiport. That is your sodium-potassium pump. Sodium-potassium pump goes in the opposite direction. Now the ratio is something you need to know for a test. It is 3 sodiums for every 2 potassiums. So let me show you how this works as a sodium-potassium antiport. 3 sodiums out for every 2 potassiums back in. So 3 sodiums out for every 2 potassiums back in. And that's an antiport. It goes against its gradients. So it goes in the opposite direction. So 3 sodiums out for every 2 potassiums back in. Then over here for vascular transport, we've got exocytosis, which is shooting stuff out of the cell. And then endocytosis is for getting things into the cell. So exo is out, endo is in. Now under endocytosis, I have phagocytosis. That's what white blood cells do. So white blood cells deal with phagocytosis. Then we have penocytosis that deals with liquids. So very, very fine packets of liquid. And then the last one is receptor-mediated endocytosis. And this one requires a receptor. So receptor-mediated endocytosis requires a receptor. Again, this chart is very worthwhile. Again, a flashcard would be nice. That way you know what goes under passive and what goes under active. Okay, now the next few slides are basically a rehash of what I've said, so I'm going to just go pass by them. This is again something I've already said, passive versus active, so I said it in the chart a while ago. I'm skipping all of these because it was mentioned. Here's that food coloring in a glass of water example I shared with you for diffusion. So these are available to you. That's why I'm clicking by them. That way you know that it's available to you. Again, this is all there. Okay, so I'm pausing a little bit. That way the video's not too long, but you can always stop the video to check out the slide. Here's an example of sodium going this way, potassium going the other way. But this is not a sodium-potassium pump. Because notice, sodium is still going what? In its normal direction. And potassium is doing what? Going in its normal direction. Sodium wants to go in, and potassium wants to go out. So this is not the sodium-potassium pump yet. This is all still passive. So we're still under passive. There's the aquaporins, the term that I've said several times in the previous videos, and now you can see the word water channel for aquaporins. So now you can actually formally see the word. It was supposed to be something you came across with because it's a general biology information, but aquaporins, the water channel. Okay, so again, this is just a rehash or more explanation of it. I do not care for all of these slides. It's available to you, but I don't care for them. Okay, this is again osmosis as a picture. It would be worthwhile studying. So this one's worthwhile. Okay. Now comes tonicity. Now this one I do want to expand on. I want to expand on these three terms at the bottom. So when you deal with osmosis and you deal with tonicity, you have isotonic, hypotonic, hypertonic solutions. In isotonic, the concentration of the solute inside the cell and the concentration outside the cell for both the solute and the water is equal. That's isotonic. Now, because the concentration of the solute is equal to inside the cell and outside the cell, and the water is also equal outside and inside, then that means that water is not going to be pulled in any one direction. Matter of fact, water is going to be pulled in what? Equally in both directions. So water is still moving, but it's moving equally into the cell as it is moving out of the cell. Okay, now we go to hypotonic. In hypotonic, the issue is that more water is going into the cell. And as more and more water goes into the cell, I call it hypo-hippo, to help you remember that. Hypo-hippo. It's going to cause the cell to swell, and we call that edema, and eventually it's going to pop, and that's going to be called lysis. So when it comes to hypotonic, water is going to go into the cell, causing edema, swelling of the cell, and eventually lysis because it will pop. The last one is hypertonic. This is the reverse situation. Water is going to leach out of the cell. So the cell is going to lose water. It's going to lose water to the environment around it. So as water comes out of the cell is going to shrivel up and shrink. So in hypertonic, water leaves, causing the cell to shrivel up and shrink, and that's going to be called crenation. Crenation, where the cell will shrivel up. Now, to show you that, we're going to see a picture. I believe there is a picture coming up. Now, this is what I just said to you a moment ago, where the inside and the outside are the same. And that's what you see in IV solutions, right? IV solutions better be isotonic. And that means there's no net movement. See how the author is really particular. It says no net movement. That's because water is going in at the same rate it's coming out. That's in isotonic. And that's what our IV solutions are. Then hypotonic is the one that I said that the cell is going to swell, causing edema, and then eventually lysis. And then hypertonic is the one that I said that water will leave the cell and cause the cell to shrivel up. So the cell will shrink, and that's called crenation. And that's under hypertonic. Now, the picture is 1,000 words here. So many books, including this one, has a picture showing you those things. So there's your isotonic that you typically find. And notice it says arrows going out, arrows going in. Arrows going in, arrows going out. That means there is movement, but there is no net movement. What goes in comes right out. No net movement. And that's your IV solution. Then hypo is the one that I said, remember hippo. So hypo, hippo, water goes in, causes the cell to swell, and eventually edema, and then lysis, it will pop. Then in hypertonic, the water will leave the cell and the cell will shrivel up, called crenation. That's why pictures are so important. Okay, then we go into active transport, which I've already mentioned by way of the chart, so I'm skipping these for that same reason. Remember, active always requires ATP, so you see it in the pictures. Okay, so it's going to require an ATP. Okay, here's our magic sodium-potassium pump. Okay, so there's our sodium-potassium pump. I can't zoom in, but if you look carefully, you'll see that the gray is the outside of the cell. The blue is the inside of the cell. And remember the directionality and the ratio I shared with you. I said 3 sodiums out for every 2 potassiums back in. Right? And this is the sodium-potassium pump. You can see where you are based upon its name. It says cytosol, so that means you're blue here. Blue is cytosol, which is inside the cell. And then the IF is interstitial fluid, which is outside the cell. So sodium, 3 sodiums out. So 3 sodiums out for every 2 potassiums back in. And you can see the potassiums going in now. So 3 sodiums out, 2 potassiums back in. That's the sodium-potassium pump. It does require ATP. Okay. And I'm going to skip on these. I'm going to leave a little bit of a timing in between my clicking so that way you can look at them if you wish to. So this is more of the same. Here's the SIM port. Oops, let me back up. Here's a SIM port showing you sodium going, in this case, in. So sodium's going in, and notice glucose is following. That's the example I shared with you when I was on the chart a moment ago. Wherever sodium goes, glucose follows. And that's called the sodium glucose SIM port because it goes in the same direction. Here I have an antiport because something comes in as something leaves. It's called antiport. Okay, then we talked about exocytosis, endocytosis. So there are pictures to give you for that one as well. There's exocytosis, where you're popping something out of the cell. Okay, then we have endocytosis, and they're going to come in 3 varieties. You have your phagocytosis. That's what white blood cells do that I mentioned when we were on the chart earlier. So that's phagocytosis. Here's another version of it. So this thing is going to be eaten up. Okay. So that's phagocytosis. Then we have penocytosis for taking in small packets of fluid and materials. Then we have receptor-mediated endocytosis. The key example that people give for receptor-mediated endocytosis is cholesterol and LDL. And it always looks like this. So if you see a picture like this, or they talk about cholesterol and LDL, then you know they're talking about receptor-mediated endocytosis. The only thing is you've got to have a receptor to make this little fold. So the receptor picks it up, the little blue sesame seeds, picks it up, folds in, and then takes it into the cell. Okay, more of the same. Again, this is a summary that many books have. Here's more of the same, summary of what books often have. You might want to use something like this to help you. So either that picture or this picture. Okay, I don't care about that. Okay, now this is important because muscles will make up most of test 2, and the nervous system is basically test 3 for us in A&P 1. So a lot of this will come back. So we have resting membrane potential. And the resting membrane potential says that there's more sodium on the outside, more potassium on the inside. There's a whole lot of protein on the inside, a whole lot of chlorine on the inside. And that means that the outside will be typically positive. So the outside's typically positive, and the inside is typically negative. And that's what you're going to see on the upcoming slide. I don't care for any of this. Don't care for this. But I do care for this picture. This picture says a lot of what I've just said, and here it is. Sodium are the purple balls. You see a whole lot of sodium on the outside. The blue ones are potassium. You see a whole lot of potassium on the inside. You see a lot of protein. All of this protein makes it a lot of negative. So that's why you see the inside being negative, the outside being positive. So at rest, if you were to do an electrical probe, like a voltmeter, something in electricity class, then guess what? You would have a charge. And the charge is positive on the outside, negative on the inside, all because it's going to be minus 70 millivolts. Now that's a number to keep in mind for an upcoming, like a future test, like when we get to the nervous system chapter, but I don't care for it right now. Just be aware that there is a sidedness and something called resting membrane potential. Why is that important? Well, when you get your little EKG done at the doctor's office and they put those little sticky things on your chest, and then after the procedure is over, they yank and pull your hair with it. Well, those sticky things that they're putting on the surface of your skin is actually monitoring this kind of electrical difference. So your skin does conduct electricity. Your cells of your body conduct electricity. That's why, even though it doesn't seem like a lot of logic, but when Frankenstein used the electricity of the lightning bolt at the top of his castle to come down and bring Frankenstein's monster to life, there was some science to that. Okay, you can actually take the little, the little paddles at the doctor's office or the emergency room and say clear and put the paddles over the person's chest and revive the person by doing a nice little jolt of electricity. Well, that's why. Okay, you can do those little EKG paddles and all that because of your skin conducts electricity, your cells of your body conduct electricity, and that's what the driving force here is. Okay, don't care. I don't care. Don't care. I've already mentioned this, the ratio. Remember, it's going to be 3 sodiums out. So 3 sodiums out for every 2 potassiums back in. Remember, they're in opposite directions and they go against the concentration gradient. That is why the sodium potassium pump is a active transport ATP required. Okay, don't care. Don't care. Do I care about this one? Not really. I don't care. Don't even care. Do I care about this one? Not really. Don't care. Don't care. Okay. This slide now starts off the flashcards that I've asked you to make. So I've asked you to make flashcards of the cell and the parts of the cell. This is where it comes in. So starting with this slide, you're going to see information that should go on your flashcard. So I'm going to click through these slides, and as I click through them, these are the things you should put on your flashcards for the parts of the cell. So here's your first slide. There's your second one, which is now on the rough ER, smooth ER story. There's more information about the smooth ER, rough ER down here. So this can go on your flashcard. That's what I mean by having flashcards. Okay, next. So that can go on your flashcard. There's more of the same without the picture. Again, for the smooth ER this time. And this is where I said that it deals with the carbohydrate and lipid metabolism. So I gave it to you earlier. There it is. So lipid and carbohydrate metabolism. This slide is of value to you. There's the Golgi. And I said it has 2 sides. That means one side is going to be the input and one side is going to be the output. And that's what I said in the picture. That's what they're talking about here. Okay, so there's the Golgi, more information. Here's the picture version, and again, you can get the same details right off the picture as well. So if you don't like the slides without the picture, you can get them right off of that. Here are the lysosome, which I said contains the digestive enzymes to break things down. That's mentioned here. So that's lysosome. There's peroxisome. Remember I told you about the hydrogen peroxide story? So this is dealing with the hydrogen peroxide story. Okay, I don't care for this one. Okay, there's more information about the endomembrane system. It is not a big deal for me because this is more of a general biology and not really hashed in A&P1. Mitochondria, again, you have information about that on for your slide. So mitochondria. Again, more information about the ribosomes and the two kinds. I said the bound ribosomes are for the rough ER, and then the free ribosomes. And notice I said that the bound ones, or the rough ER, is eventually going to be for what? Export. So when I was on the picture earlier, I said bound ribosomes are on rough ER, and they're making proteins for export, whereas free ribosomes are for making proteins for the cell's internal use. And that's what I was summarizing earlier. Okay, more information about ribosomes and the protein synthesis story. Cytoskeleton, and I've mentioned microfilaments and microtubules when I spoke about cytoskeleton. So microfilaments, microtubules. Okay, so microfilaments, more mentioned there. I don't care about that one. Microtubules, again, more mentioned there. So again, they're part of the cytoskeleton. There it is again, cytoskeleton, giving structure and framework to the cell. There's centrosome, and I mentioned centrioles, and I told you that centrioles are part of cell division. They actually make the spindle fibers for the chromosomes to separate on. So if you've ever seen a picture of mitosis, metaphase, anaphase, telophase, well, that cell division requires centrioles to form spindle fibers where the chromosomes can be separated on. Okay, so here's an arrangement of those. And again, more information down here. Again, there's my magic phrase I mentioned a second ago, the mitotic spindle. So that's mentioned at the bottom. Proteasomes I don't care too much about, but it's there. Okay, don't care too much about the cilia and flagella. Just know that they're there, and not too much will be asked about that. Just be aware that they're there. So cilia, flagella. Here they are again. You'll see these kinds of things in your intestines for A&P2. Okay, now those are the things you need to make flashcards for when it comes to this chapter. If you don't make the flashcards, it's going to be tough to remember what's what. Then finishing up this chapter, we have something called membrane junctions. We have tight junction, desmosomes, and gap junctions. Now, tight junctions are found in the intestines, in your gut. So tight junctions are found in your gut. They're making sure that things that are outside of your gut stay outside the gut and not get into the gut. So if you actually had a leak, a tear, so that the stuff outside your gut, the liquid outside your gut, got into your gut, you would have a heavy-duty diarrhea problem. And that's what happens when you get food poisoning from Shigella or Salmonella. It actually tears at these tight junctions, and now you have a nice little leak that allows fluids of your body to get into your intestines, and then you're going to have a heavy diarrhea even if you don't drink anything. So even if nothing's going down your food pipe, no liquids going down, nothing, you're still going to have diarrhea because your body fluid will be going through your intestines and out your body. So you'll be basically leaking fluids out through diarrhea this way. So tight junctions are keeping your things like the intestines very, very tightly packed because the cells are really joined close together. So tight junctions. That's how one way we join cells together, tight junctions. The next one is desmosomes. I call these snap buttons. Snap buttons. Okay, they're like two little rivets going together, or snap buttons. That's how we hold one cell to another cell joined together with little snap buttons. Some people call it Velcro or whatever, snap buttons. Then we have gap junctions. Gap junctions are found in the heart. So gap junctions are found in the heart. Gap junctions have little tubes in them. These cells have tubes in them that allow for an electrical ionic connection between one cell and another cell. So one cell has a little pipe or a tube connected to another cell. So one cell has a pipe or a tube connected to a neighboring cell. Down that tube, we have ions going across, like sodium, potassium, things like that. Ions going across. That means that when I fire cell A, then soon after, because the ions are going through that tube connected to cell B will fire shortly thereafter. That's why gap junctions are typically used in the heart, allowing one cell of the heart to communicate to a neighboring cell of the heart. Okay, let's go look at this from the author's point of view now. So here are the tight junctions. And you see that they're really, really tightly packed together to prevent any kind of fluid leak. So like in this case, they're using urine, but you could use digestive system as well. Very, very tightly packed. Okay, then we have desmosomes, which I said were like snap buttons. Okay, where they hold them tightly together with rivets. And then the last one was gap junctions. And this is where I said that one cell communicates to a neighboring cell by way of a passageway that allows ions to move across. So it allows one cell of the heart to communicate with a neighboring cell of the heart. You'll see this come up again in A&P 2, so you want to know it now. So gap junctions, dealing with the heart muscle, allowing cell-to-cell electrical communication. Okay, next slide. Okay, here is a picture version. And this is a very good thing because what I described just now often doesn't work. So here is a tight junction. So tightly packed that water cannot get between the cells. So cell A is on the left, cell B is on the right, and this right here is so tightly packed together that water cannot go between the two cells. That's why it's called tight junction. You can't get this water here to go between the cells. Then the desmosomes are this thing. They look like Velcro or snap buttons that hold one cell joined to another. You know how some men's pants, jeans have button fly? Think of it like that. It's like the button fly. They're snap buttons. Then the last one was gap junctions, where you see these little tubes. So cell A is where the cursor is bouncing on the left. Cell B is over here on the right. And this tube has ions passing through it. These tubes have ions passing that allow cell A to stimulate cell B. So those are your membrane junctions. Okay, don't care for that. I really don't care for that. I do care for this. You do need to know what's inside of the nucleolus. The odds of that question being asked are slim, but they may get a chance to be asked because it's a slim chance, but you should at least know it. So nucleolus contains the RNA part. Okay? That's what makes the ribosomes. So nucleolus will deal with the ribosomes. Again, not as a big of a question, but if they ask it, that's what they'll ask about. So nucleolus dealing with RNA, and that's where the ribosomes are made. So nucleolus, ribosomes. So if they ask, that's what they're likely to ask. Okay, don't want to care for this picture. I do need to know this word here, chromatin. Chromatin is loose genetic material when the cell is not dividing. So when the cell is not dividing, the loose genetic material is called chromatin. That you do need to know. So loose genetic material when the cell is not dividing. What do you think it becomes when the cell does divide? It becomes chromosomes. So do you see the difference? When the cell is not dividing, we call that genetic material chromatin. And when the cell is dividing, then we call that chromosome. That's the difference. Okay. All right, DNA. This is a rehash from another chapter, so I'm going to pass this by. We've already seen this DNA story earlier. Remember I said that was the most commonly missed question? Nucleotide is the basic building block of DNA, and a nucleotide has your 5-carbon sugar, your phosphate group, so your pentose sugar, phosphate group, and your 4 nitrogenous bases. Like I said, that was the most commonly missed question almost always. So nucleotides, basic building blocks, they're made out of 5-carbon or pentose sugar, phosphate group, and the 4 nitrogenous bases. Okay, don't care. I already told you. Actually, I don't care for that. Chromatin, I've already shared with you. I said that when the cell is not dividing, so when the cell is not dividing, the genetic material is called chromatin. When the cell is dividing, the genetic material is now called chromosome. And so when you're in words like prophase, metaphase, anaphase, telophase, during cell division, during the actual mitosis, then we go from chromatin and change it into chromosome. When the cell is finally divided and everything, the chromosome goes back to chromatin. Here is a picture showing you this is what it looks like when it's unraveled, when it's not dividing, chromatin. And when the cell is ready to divide during cell division, it becomes chromosome. And we've already seen this storyline earlier. I told you A and T has 2 hydrogen bonds. G and C has 3 hydrogen bonds. That was previously mentioned in another chapter. I also mentioned that A and T go together for DNA. A and U would go together for RNA. And G and C always go together for RNA and DNA. I don't care for this. Do I? Not really. No, I don't. This part right here is a gene. So a sequence of DNA that codes for a protein, that's a gene. So a sequence of DNA, I'll say that again. A sequence of DNA that codes for a protein is called a gene. Simple definition question. I don't think they're ever going to ask you about this stuff right here. But they will ask you for the definition of a gene. So a gene is a sequence of DNA that codes for a protein. And it should say specific protein to be more accurate, because it does code for a specific protein. Okay, and there's your question why I said you need to know that. What is a gene? A gene is a sequence of DNA that codes for a specific protein. Okay, now I'm on this slide here, and it says protein synthesis. Okay, I have a video supplement ready for you for protein synthesis called Transcription and Translation. I want you to watch that video instead. So when you go to the lecture video series, you will find Chapter 4, and then right below that you'll find Chapter 4 Supplement, which has a Part 1 and a Part 2. Make sure you watch both of those. All right, so that took care of the protein synthesis. Clap, 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 stop. Okay, now we're on cell division. So cell division. You have your mitosis, you have your meiosis, your cell division. You will need to know the difference between mitosis and meiosis. So you will need to know the difference between mitosis and meiosis. Okay. We've already talked about chromatin and chromosome earlier, so that's done. Okay. This is where I would start putting things on flashcards. So you will need to know the cell cycle, which I'm going to go over by way of a picture. So I'm skipping all this because I'll be describing a picture. And that picture is this cell cycle here at the top. Okay. So the cell cycle at the top. I do not care for the DNA replication part. I care for the cell cycle. So here I have where the first box is, where the cursor is, that is G1. That's where we have growth 1. Then I have S. S is for DNA replication. And then I have G2. And then I have the actual cell division, in this case, mitosis. So if I were to look at only the blue, I have G1, I have S, and I have G2. That makes up interphase. So that is going to make up interphase, everything in blue. Then down here would be the mitosis. Now G1 is growth 1, getting all the materials ready that I need for S. S is where DNA replication occurs. And then G2 is getting ready for all the materials necessary for the actual mitosis. So most of the cell's time is spent in interphase. So most of the cell's time is spent in interphase. Now the things I've said just now are actually found on this slide and this slide. So that's what I just went over. I basically went over this slide, and this slide by way of the picture. Okay. All right, moving on. So here's our G2. That's also covered. And now we have our interphase. Now starting from this slide onwards, make sure you make flashcards for these. So I'd draw a rough sketch like the one on the top, and then the information on the back side of the flashcard. Same is true for the actual phases of mitosis. You have the 4 phases of mitosis. Well, the author has given each of them. There's the paragraph form for prophase. There's the picture and the stuff that goes on your flashcard for it. Then there's metaphase, picture, and the flashcard. Anaphase, picture, flashcard. Telophase, picture, flashcard information. So make sure you make flashcards for the cell cycle. There's cytokinesis, also mentioned here. That's important. And here's a better picture of the cell cycle as well. So G1 is basic growth. S is where DNA replication occurs. G2 is where the growth and necessary materials for the cell division. Interphase is where most of the cell spends its time. That means the genetic material, since the cell is not dividing in this blue area, since the cell is not dividing during the blue, that means that the genetic material must be called chromatin. So during interphase, the genetic material will be called chromatin. Only during this stage in pink down here will chromatin turn into chromosome. And when we're done with mitosis, it will go back to being chromatin again. Okay, that's important. Okay, last few things. Okay, cell death and aging, called apoptosis. And that is this term right here, by the way. Apoptosis is pre-programmed cell death. So apoptosis is pre-programmed cell death. Okay? So every cell is pretty much destined to die. It has a certain shelf life. So apoptosis, pre-programmed cell death. And that's what this whole paragraph here is talking about. It's apoptosis, pre-programmed cell death. Okay, there's your word again. Programmed cell death called apoptosis. So it's pre-programmed. Okay, I don't care too much about this at all. Don't care about this either. I do want you to know the definition I gave you. Apoptosis is programmed cell death. That's important, but I don't care for the rest. I don't care for this slide. Don't care. And that ends our slide.